In this chapter, we're going to take a look at metabolism when we talk about energy, and in particular how enzymes influence energy. We will look at the forms of energy and talk about the laws of thermodynamics when we discuss kinetic and potential energy as well. Look at metabolic reactions and how ATP is involved in metabolic reactions. And mostly we'll look at metabolic pathways, talking particularly about enzymes and how enzymes affect the energy of activation. And we'll discuss the flow of energy, previewing the next few chapters on photosynthesis and cellular respiration. And notice energy coming down from the sun, being given off as heat, driving photosynthesis, the animal eating it. Notice that energy has this flow to it. It is not, however, cycling back. Energy always flows. Energy does not cycle. It's one of the prime concepts behind the ideas about energy. So what is energy? Well, it's our ability to do work. It's the ability to bring about some kind of change. We're changing this solar energy into chemical energy of the plant. The animal is taking in the chemical energy and changing it into mechanical. It's about taking this energy, converting it, changing it, and doing work with that in a flowing pattern. We have two forms of energy. We have kinetic, or the energy of motion. This is sometimes called our mechanical energy. The ability to walk around, our muscles to move, water flowing down a river, all of that, your car riding down the road, that's all examples of kinetic energy, energy of motion. Potential energy is stored energy. We sometimes refer to this as chemical energy. The gas sitting in the tank of your car that is parked is potential energy. The food that we ingest is potential energy. A ball sitting still on the ground is potential energy. If you walk up and kick the ball, now the ball has kinetic energy, energy of motion. All of our energy is ultimately dependent upon the sun. Solar energy drives all of these processes. Let's talk about the laws of thermodynamics. I'm on the top of page 102. We'll start with the first law, sometimes called the law of conservation of energy. These two laws are going to help us explain why the energy can flow and not cycle back. The first law says energy cannot be created nor destroyed, but we can change it from one form to another form. We can change chemical energy to mechanical energy. We can change kinetic energy to potential energy, but we cannot create it and we cannot destroy it. We can only change its form. The second law is sometimes referred to as the law of entropy. Entropy means to turn inward. It says that when you change this energy from one form to another, from chemical to mechanical, that there will be a loss of usable energy. When we turn the carbohydrates that we eat into ATP, we lose some of that energy as heat. When our muscles convert those carbohydrates into usable energy, we lose some of that as heat. This waste energy that we lose, this heat, goes to increase the disorder of the universe. It makes the universe a little less organized and a little more disordered. However, your book does talk about the fact that just because we have become less organized and more disordered, we have achieved more stability. Think about a dirty room. A dirty room, although dirty and not organized, is very stable. You can move a piece of something from one to another and not really notice any more disorganization. It's fairly stable. However, a clean room, a very organized room, is not a very stable room because it is very easy to disrupt the order. You can move one single thing out of place and now you have all this disorder. So although we are less organized, we are more stable. You can see an example on page 103 where we have our very organized glucose molecule. It has a lot of stored potential energy, but it's not very stable. It would not take very much to disrupt this glucose molecule. When we break this down into carbon dioxide and water, well, now we're not as organized and we don't have as much potential any longer. However, we've achieved more entropy, more stability, because we have broken it into smaller parts. I want to move and talk about your metabolic reactions and your energy transformation. We're going to move over to page 104 in your textbook. I'm going to start by talking about metabolism. The sum of all chemical reactions that occur in our body is metabolism. Now, we can show chemical reactions by using a chemical equation. We can say that A plus B produces C. In this case, A and B that are participating in the reaction are called our reactants. Our reactants occur on the left side of the arrow. On the right side of our arrow, we have our products. In this case, we only have one product. We could have many. They're the result of the reaction. After the chemical reaction has occurred, what are we left with? We can turn this into any. We can, for example, break down the sugar molecule. And sugar can break down into carbon dioxide and water. And we'll have a little energy or ATP left. We can balance this. At this point, this is an unbalanced chemical reaction, but we could balance it. This is considered our input or our reactants. Notice we only have one reactant, and in this case we have two products, the result. When we undergo a chemical reaction, there's going to be some energy that is available, because anytime you convert energy from one form to another, we're going to lose some energy. We're also going to create what we call free energy. We represent free energy with a capital G for free energy. This is the amount of energy that's going to be available to do work. We're going to be able to use to move our muscles, to walk around. We'll see two kinds of reactions. We have our exergonic. Recall from an earlier chapter, exo refers to out, ender refers to in. In an exergonic reaction, like cellular respiration, our products have less free energy than the reactants. In an endergonic reaction, the products have more free energy than the reactants. This is because an endergonic reaction requires the input, endergonic, N, of energy. Requires energy to be input. So if we're going to put in energy to this reaction, then logically your products will have more because we've put energy into the reaction. In an exergonic, look at where the energy is going. The energy is going out, so the products will therefore have less energy. So what is this energy? To talk about energy, we have to talk about ATP. ATP is going to be the molecule that's going to actually carry around the energy in our chemical bonds. It's the common energy currency for the body. It is the money we must spend in order to do any type of work in the body. So this molecule that can carry around high volumes of energy is properly called adenosine triphosphate. And this is a very important part of the name. This tri tells us that we have 3 phosphate molecules along with that nucleotide adenosine, adenine. We can constantly make ATP from adenosine diphosphate, which carries only 2 phosphates. This is going to happen by the reaction that we referred to earlier in a chapter called hydrolysis. Remember that lysis means to split, and hydro refers to water. This is using a water molecule to split up a polymer into its smaller parts or monomers. So why is ATP such an important common energy currency? Because it has these 3 advantages. One, it is common. Every cell of every organism on the planet can use ATP. It does provide in this hydrolysis reaction a sufficient amount of energy. Notice sufficient here. That is an important term. Sufficient means just enough. Not in excess, not too little, just enough. What this does is provides us enough energy to do our work without wasting energy. ATP can also be coupled with an endergonic reaction to minimize the energy loss. The production of ATP is exergonic. Exer, energy is going out. An endergonic reaction, one that needs energy put in, can capture this energy being given off from the production of ATP and use it for another process. Therefore, energy is not really lost out into the environment. Now remember, we can't destroy energy or get rid of it, but we mean lost out into the environment. So the production of ATP, an exergonic reaction, can be coupled with an endergonic reaction in order to minimize our energy loss. Three very important advantages to using ATP. Looking at the basic components to ATP, it's made of adenine, one of our nucleotides, and ribose, which is one of our pentose sugars. Together we call it adenosine. It's also made of 3 phosphate groups, and these phosphate groups are very easily removed. That's what makes it such a good exergonic reaction. We do want to always couple this reaction so that in breaking down ATP into ADP, remember this is diphosphate for 2, this is tri for 3. Here are the 2 and here's the 3rd phosphate. In breaking the ATP down into ADP and phosphate and giving off that energy, this endergonic reaction below here can use this energy to drive its process. By coupling them together, we have very little wasted energy. An example in your textbook on the bottom of 105, if you look carefully at A, it tells you that ATP breakdown is exergonic. Energy is given off. And muscle contraction is endergonic. Energy must come in. We take this energy being given off, we pump it into our muscle contraction and get movement out of that with very little wasted energy. So why do we have to have ATP? Primarily we have to have ATP to do our cellular work, to do all the things a cell needs to do, to make ribosomes, to make proteins, to create energy, to copy DNA, to go through basic cellular jobs. We can do 3 things: chemical work, which is the energy we need to make our macromolecules like our carbohydrates and our proteins, our lipids. ATP supplies that energy. ATP also supplies the energy we need to pump substances around across the plasma membrane. If a substance is not of the right size or the right ionic charge, then energy must be expended to move it across the membrane. ATP also gives us the energy to do our mechanical work, our energy to contract our muscles, to walk around, for the flagella of different cells to beat, to move. We need energy for that mechanical work. ATP is that immediate energy that we must have for these processes to occur. All right, the third major topic of Chapter 6 begins on page 106. We want to talk about our metabolic pathways and particularly our enzymes. Now, we've previously looked in this chapter at what a chemical reaction is. It begins with a reactant and it produces a known product. Now, in a metabolic pathway, we're talking about reactions that can occur in a sequence, where the product of an earlier reaction can become the reactant of a later one. Now, by using this series and capturing this energy, we can use the energy in very small increments. So, let's look at a metabolic pathway. We start with a particular reactant. That reactant will make a product. Now, that product, B, is now going to become the reactant of the next part to make the product C. C becomes the reactant to make the product D. D becomes the reactant to make the product E. E becomes the reactant to make the product F. And F becomes the reactant to make the product G. In the middle, all of these reactants turning to products are referred to as intermediates. Whereas A is the initial reactant, the one we're starting with. G is the end product. But all along the way, we have these intermediates. Whether they are reactants or products, all depends on which portion of the pathway you are looking at. So let's look at just one particular portion of the pathway. Let's clear a little bit of this. And let's only look at the portion of the pathway, narrow a little more. Let's look at only the portion of the pathway here. Now, in this highlighted portion of the pathway, C would be considered a reactant, and D would be considered a product. Those terms will change depending on what part of this reaction I'm looking at. Enzymes are going to be particular proteins. They're going to act as a catalyst. Now, the job of a catalyst is to speed a chemical reaction. The unique thing about an enzyme is that an enzyme does not become a part of the reaction and is not used up by the reaction. So because an enzyme is not used up by the reaction itself, we only have to use a little small amount of them. Using the small amount, they'll go into the reaction, they'll speed the reaction. At the end of the reaction, they will come out unchanged. To name an enzyme, we add the suffix ase to the end of something called a substrate. Substrate is just a specialized term for reactants. But we only use the term substrate if our reactants are a part of an enzymatic reaction. For example, here is the thing we were looking at earlier, where reactant A made product B, B became the reactant to make C, C became the reactant to make D, and so on. Well, notice now above the arrow we're showing E1, E2. This indicates that this is a reaction that is sped up by the use of an enzyme. Because we have now used an enzyme, instead of calling A a reactant, we call A substrate. Substrate tells me that this chemical reaction will utilize enzymes. So if I want to name my enzyme, I simply want to add ASE to the end of the name of my substrate. Enzymes are very specific. They only accelerate a particular reaction. And for an entire metabolic pathway to occur, you will have to have every unique, every specific enzyme. Because if you were to remove any one of these enzymes, the reaction would stop. The only way to make product G is to not only have all substrates present, but to also have all enzymes present. The end product will fail to appear without all enzymes present. Notice, not only must they be present, they must be functional. If you'll think back to a prior chapter where we were talking about denatured, enzymes are proteins. And if proteins lose their basic shape, whether that's primary or secondary or tertiary, if they lose their basic shape, they lose their function. So for an enzyme to be present is not enough. It must be present and it must have the proper shape, therefore have the proper function. So an enzyme must be present and an enzyme must be functional in order to work and speed up a chemical reaction. Enzymes work because they have the ability to lower what is called the energy of activation. So think about the term activation. That means to start. Energy of activation is how much energy do I have to add to this reaction to start this reaction? Now the energy could be heat, the energy could be many things. How much energy do I need to add to start this chemical reaction? An enzyme assists the reaction by lowering the amount of energy that's needed. Think about making tea. If you are served unsweet tea, and you choose to sweeten that tea, and you add sugar to the tea, if you just dump the sugar in the tea and look at the tea, you're going to just have unsweet tea with a glob of sugar at the bottom. But if you were to stick a spoon down inside of that and stir the tea, your spoon is acting as an enzyme. It is speeding up the reaction, but it is not becoming a part of the reaction. Because when you're through dissolving your sugar, you'll simply remove your spoon out. The spoon is unchanged. You've sped up the reaction. The stirring by the spoon has lowered the amount of energy needed to cause that reaction of that sugar dissolving in that tea. So what could you do if you didn't have the spoon? Well, then we'd have to look at other options. We might choose to heat the tea and so on. We'll look at those other options shortly. Let's talk about the enzyme-substrate complex. We're talking about the complex between the enzyme itself and the reactant in an enzyme reaction. Remember that substrate is simply another term for the reactant of an enzymatic reaction. Now, for the enzyme and the substrate to combine together, the enzyme can only join to an area that's referred to as the active site. Enzymes, remember, are specific. An enzyme is only for a particular substrate. That's why we name the enzyme after the substrate. The active site can actually undergo a change in shape to accommodate its particular substrate. We call this the induced fit model. The shape change is going to force that substrate and enzyme to bond together and work to speed up the reaction. When the reaction is over and the enzyme is removed, the substrate will return back to its original shape. We're going to look at the factors affecting enzyme activity. And the first factor will be the concentration of substrate. Now remember that substrate is the name of a reactant where an enzyme is being used. So enzyme activity will increase when there's more reactant available, when there's more substrate available. Because when there's more substrate, then you have more collisions. There are more reactants at play. There are more collisions. Therefore, the enzyme is working and increasing the reaction. How much enzyme is present can also increase or limit the rate of the reaction. So if we want a reaction to go at its fastest, we want to make sure that there's enough substrate available. pH is also a factor when you're talking about the speed of an enzyme, because most enzymes have a particular pH. If it is an enzyme that only works at a pH of 4, and you have placed it in a solution with a pH of 7, in order for that enzyme to work, you're going to have to lower the pH of your solution. When you lower the pH of the solution to its optimal pH, then it will work, because otherwise a change in the pH changes the shape of the enzyme. And when you change the shape of the enzyme, as changing the shape of anything with science, then the structure no longer functions as it should. Temperature is going to affect the rate because the activity of an enzyme increases as temperature increases. It's just like living individuals. If you're a living individual, when it's warmer, you're more active. When it's cold, you're less active. Enzymes behave the same way. Warmer temperatures cause more effective collisions between the enzyme and the substrate. But there's a delicate balance. You get the temperature too hot, and it will actually destroy the enzyme because it will change the shape of the enzyme. We say the enzyme is denatured at that point. Denature just means to lose its normal shape, denature, without the natural shape. Typically, the two ways to denature the enzyme involve putting it in the improper pH or elevating the temperature to too high of a level so that the protein, which is what an enzyme is, breaks down. We also want to take a look at our organelles and how they deal with the flow of energy. We talked earlier in the lecture about the flow of energy coming from the sun, creating photosynthesis, organisms ingesting those plants. Well, we want to look back one more time at these two organelles we covered in the cell chapter. We do have chloroplast, which is the organelle in which photosynthesis is going to take place. We're going to capture the solar energy and produce a carbohydrate out of it. Now, animals do not have chloroplast. We can't capture solar energy and make a carbohydrate. We do, however, have mitochondria. So does a plant. We can take in that carbohydrate and turn it into ATP. Both of these organelles work through a process called oxidation and a process called reduction. So we need to stop and look at what oxidation and reduction is. Sometimes we'll call these redox reactions for short. And what's happening is we're passing electrons. Electrons are passing from one molecule to another molecule. And remember that electrons are just little bundles of energy. And as we pass them around, energy is given off and can be picked up. If a molecule loses an electron, we say that it has become oxidized. Now, giving away electron, remember, is going to make it a positive ion, but we say it has become oxidized. Now, a molecule that gains an electron, we said in previous chapters that would make it a negative ion, that is termed reduced. Now, these take place simultaneously. At the moment a molecule loses an electron, there's another molecule to gain that, because you don't want electrons just moving around in an unchaperoned pattern. Electrons that aren't chaperoned, electrons that are just free and bouncing around, are giving off tons of energy, and that energy can be destructive. So we want to make sure that as one molecule is being oxidized, losing electrons, there is an opposing molecule ready to gain those electrons. These are the processes that drive photosynthesis and cellular respiration. It's the creation of energy because we have oxidation reduction. We have the moving of those electrons, the losing and the gaining of them.
Easy way to remember that is to remember that Leo, as in Leo the lion, says ger, where each letter stands for something. To lose an electron is to be oxidized. To gain an electron is to be reduced. Just a little tip to help you remember the difference in oxidation and reduction.
So we looked at the forms of energy. We talked about the differences in kinetic versus potential, the energy of motion, kinetic, the energy of potential, the stored energy. We looked at the two laws of thermodynamics and talked about entropy. We looked at metabolic reactions and how ATP plays a role. And we looked at enzymes, particularly how an enzyme makes the reactant, now called a substrate, how it accelerates the reactions, and the factors that will influence the way an enzyme works. We want to make sure that pH is correct, temperature is correct, concentration is correct, cofactors are present, and so on. We know that there is a flow of energy, and that in that flow of energy, it's particularly involving the moving of electrons, and we're going to use those moving electrons to drive the next two processes we'll talk about, cellular respiration and photosynthesis.